Hey guys, it's Mrs. Couch here, and we're going to talk about plasma membranes and membrane permeability today. Okay, so you're probably going to have to stop and write some stuff down as we're going through this, or maybe even rewind. And I'm going to bring up a couple of videos and some pictures to help you understand some stuff too. So here we go. All right, so we're looking at the cell membrane and the membrane proteins and all the phospholipids that make up the membrane. Okay, and we're going to look at the permeability of the membrane as well. Okay, so the plasma membrane is mainly, hopefully you remember this, mostly made of a phospholipid molecule that looks kind of like this. It looks like a tadpole with two wackadoodle tails here on the end. Okay, so this top part is our phosphate group. And there's also this glycerol part that's also attached. Okay, I don't think that you would need to know like the whole thing of like each part, but as long as you know this is the phospholipid head. Okay, so this is where our the phosphate group is, and then there's some glycerol, because remember, fats are made up of glycerol backbones and fatty acid tails. Okay, and then we have these fatty acid tails, which are like the tails of our tadpole that are hanging down here. And they can be saturated or unsaturated. So remember, this is going to be important later. If it's saturated, it's going to kind of look like this with no double bonds, and these would be more rigid and less fluid. And if it's unsaturated, it has double bonds in those tails, okay, in those carbon chains, and that's going to make it more fluid and movable. And you can see it has a little kink in it already. Okay, the heads, the phospholipid heads, are polar or hydrophilic, and the tails, because they're made up of fats, are hydrophobic and are nonpolar. Okay, so when you look at the arrangement of the plasma membrane, let me get everything on there. Okay, it is a bilayer. So there are two layers these with the, like this is the outside of the cell membrane, and this is the inside of the cell membrane. The heads are arranged on the outside parts, like a sandwich, like the outside parts of bread, and then the inside would be your jam and all your other stuff that's going on in here if you have a jam sandwich. Okay, so these are our tails. So this is kind of what it's set up like. Okay, so it's primary, again, made up of phospholipids. Okay, and phospholipids are called amylopathic. This is just a fancy vocab word that says it means that they are molecules that are both hydrophilic and hydrophobic. They have have a part that is polar and a part that is nonpolar, and that's what allows us to form this bilayer. Now, if you notice, there's some other junk that's in here. These are all really important things. I just called them junk, but they're really, really important. We're going to talk about those in just a few minutes. Okay, so the plasma membrane is selectively permeable. It only allows certain things to move in and out. So because the heads are hydrophilic and they're orientated to the outside, anything that is also hydrophilic would be attracted to this. So water is very much attracted to this. Okay, it's polar. Anything that is polar would be attracted. And the tails, again, are hydrophobic, so they're facing away. So they're on the inside. Okay, we talk about the this being like a fluid mosaic model because these little pieces don't really stay stagnant in place. They move, okay, and they work together, and the temperature affects the fluidity of the membrane. Okay, so if it's warm, these guys are moving around pretty fast. They kind of stick together, but they can like actually kind of fall apart if it gets too hot. And if it's cold, if you're in a cold environment, they don't move a lot. And that's also not super great because it isn't allowing things to move in and out. So in areas where it's really cold, you would expect to see organisms have evolved to have a lot of these unsaturated tails, okay, with all these kinks in it that allows it to keep moving, because if they were all saturated, they would be like little sticks that are stuck in place and they're not going to be able to move. So the unsaturated tails help to increase fluidity and help keep the cell membrane moving. Also, this molecule right here, which is cholesterol, which is naturally made by animals, helps to keep it fluid. Okay, so we'll look at that as well. Just a second. Okay, so the kink tails, I hope you can see that, helps to keep these, like the unsaturated tails help to keep it moving, to allow things to move in and out, because that's its job. right, I'm going to show you real quick this little video. Make sure I'm on the right one that I want to show you. Yeah, let me show you this and then I'll come back. All right, so here's this little video about it. This is the right one. The guy's voice is a little weird, sorry.
Between the living machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life rests on this veil of material. A supple membrane, just 2 molecules thick.
Okay. All right, so that's kind of what it looks like, and just to kind of give you that idea of it's not stagnant, it is moving. And you could see in the video too, it looked like they had a lot of unsaturated fats in the tails. I don't know if you noticed that, but there were a few that were unsaturated that allowed for that movement. Okay, they also mentioned some of these other molecules, okay, like cholesterol. And cholesterol, again, is naturally produced by animals. It's not produced by plants at all. And it's this little molecule right here that you'll see tucked inside, which also helps to keep these things from kinking up. And it helps to keep fluidity at high and low temperatures. Okay, again, high temperatures reduces the movement, and it can actually cause it not anything to get through because it kind of denatures things. And then low temperatures also cause the tight packing of the phospholipids. So having like this little spacer in there kind of cholesterol helps to keep the phospholipid bilayer moving. Okay, and again, it's called a mosaic because it looks like a mosaic. It's one big thing comprised of many little moving parts. Oh, arrows. All right, then there's membrane proteins. Okay, there's 2 different types of membrane proteins. So this is the second molecule that proteins are the second macromolecule that are really important in making the cell membrane. There are integral proteins and peripheral proteins. Okay, so integral proteins are, the word integral means it's like embedded, or it's a necessary part that's like part of the bilayer. Okay, so integral, it's actually a part of it. Okay, and these would be the transmembrane proteins, like these guys, which we talked about when we talked about sodium potassium pumps and protein channels for facilitated diffusion and all those kinds of things. So they're actually like stuck in between these membranes. And when you saw the video, you saw they actually kind of move around in the membranes because it's fluid. Okay, and then there's also proteins that are peripheral, which just means if it's on the periphery or it's on the outside, okay, like these glycoproteins or this glycolipid thingy out here, okay, this is not a protein, but it's on the outside and it kind of floats around and moves. And these glycoproteins are really important in cell recognition and your immune response. Okay, so they're just loosely bonded to the protein, or to the phospholipid bilayer. Okay, so here's just some more details about cell recognition, which I was just talking about. So you have these little channels, and I think are these little flags, and I definitely mentioned this in 10th grade. I don't know if you remember it, it's been a while, but these are actually like antigens that, that's what these guys are, and they help cells to recognize each other. So your immune cells, your white blood cells, are constantly floating around and patrolling for things that are foreign invaders. And you have like this barcode or this specific glycoprotein code on the outside of your cells that your cells recognize them being your own. Okay, and so if it's there and it matches what your antibody or your antibodies and your white blood cells recognize as important and they should be there, they don't attack it. But if they don't match, then there can be a problem. Okay, and so they can attack it. And sometimes weird things happen where your body attacks its own cells when you have an autoimmune disorder, like arthritis, some types of arthritis and lupus and those kinds of things. So these are just little flags. We'll talk more about them in class that like pull certain things into the cell. All right, so here's a crazy question. Okay, I had to look up how to even say this word, but it's Chenaphidia. I didn't say it very good. I had it down before I started the video. But anyway, this fish is also known as the crocodile ice fish. It lives in the Southern Ocean around Antarctica. These waters are known to maintain extremely low temperatures all year, ranging from approximately 28 degrees Fahrenheit to 35. So it's super cold. Okay, around freezing. These fish have adaptations that will allow them to live in the cold and low oxygen waters. Remember, if it's cold, there isn't a lot of oxygen probably either. Okay, so if you were to examine the membrane lipid composition of cells of this crocodile fish, what would you expect to find? You can pause and write it down if you want. Okay, and justify why you might find this. So you can pause and write it down. And if you did, and you've unpaused now and you wrote something down, like you should be thinking this to yourself, I would expect to see a lot of unsaturated fatty tails in my cell membranes of these organisms instead of saturated, because that allows for the membranes to stay fluid in these super cold temperatures. I would also expect those little suckers to be very high in cholesterol, because cholesterol also helps to keep the cell membranes moving in cold temperatures and just moving in general. So that's what I think you should have down for that one. We'll talk about it in class. All right, last little bit, plant cells. Okay, so let me bring out this stuff, because we just did a whole bunch of stuff about organelles, that there is a cell wall, which these dark thing like around here, these are cell walls. Okay, they help to cover the cell membranes. They are not found in animal cells, but they're also found in fungus, and bacteria have them as well. But in plants, they're made up of cellulose, which we learned in our unit 1. These cell walls are very thick, okay, to help give the cell structure because of the arrangement of the glucoses that are in there. But they also contain little teeny tiny holes called plasmodesmata, which are little channels between one cell to the next, okay? And the vacuoles inside the cell can hold a lot of water that helps to increase the pressure inside the cell, and actually keeps the cell walls actually helps to keep the cell membrane from bursting. So plasmodesmata are really important in regulating the fluid movement in plant cells, and we're going to see some of that in class. All right, so that's it for cell membrane and fluidity. I hope this was helpful, and we'll be doing some more stuff with this in class. We got a bubble lab. It's going to be fun. All right, talk to you soon. Bye.